Hi everyone. If you missed class this week, I just wanted to go over the sliding filament notes that are uploaded in the module. They will open in Kami. So remember that Kami will allow us to annotate the notes. The ones I'm showing you right now are already completed, and I'm going to just go over the steps. So you can start and pause the video as you are color coding and annotating. And at the end, you should have something that looks similar to what I have on the screen right now. All right, so just a review from last week. If you remember, in the sarcomere, we have a thick filament called myosin and a thin filament called actin. Okay, they are both protein filaments, and if they are not connected, contraction cannot happen. So we have our myosin down here. Okay, I color-coded that in orange. Click off of that. And then we have our actin up here that is color-coded in that teal color. So we have the thin filament of actin and the thick filament of myosin. Now above the sarcomeres, we have the rest of the muscle tissue. So we have a motor neuron that is going to be in charge of sending the impulses from the nervous system into the muscle. So the motor neuron is making contact with the sarcolemma. So the sarcolemma is this long horizontal area that wraps the muscle tissue. The sarcolemma has tubes, passageways that go down vertically into the muscle called T-tubules. They're transverse tubules, so they are perpendicular to the sarcolemma. And then we have the sarcoplasmic reticulum. Part of it is pictured here. It's not fully pictured here. And this will run completely through the muscle tissue underneath the sarcolemma. The sarcoplasmic reticulum is like the endoplasmic reticulum in other cells. It's in charge of transport and storage, but sarcoplasmic just tells you that it's in a muscle cell. So anytime you hear sarco, you should think muscle. So sarcoplasmic reticulum, the sarcolemma, they are part of the muscle cell. So those are our structures. We have our myosin, we have our actin, we have the motor neuron making contact with the sarcolemma. The T-tubule runs down from the sarcolemma into the sarcoplasmic reticulum. Right? So now we're going to look at the chemical processes, the physiology that happens. So the motor neuron has these vesicles, which are areas that hold neurotransmitters. The neurotransmitter we're working with in this function is acetylcholine. So the acetylcholine is shown as the triangle shapes right here. So as the impulse comes from the brain, the motor neuron receives the signal and it releases acetylcholine from the vesicles. The acetylcholine will then travel into the sarcolemma and will attach to sodium channels. These sodium channels, when they bind with the acetylcholine, they open, and as their name implies, they release sodium ions. So now we have a charge in the cell, that creates an action potential. So the sodium channels create the charge, the action potential that runs through the muscle, down the T-tubule. And the sarcoplasmic reticulum gets that action potential, and it triggers the sarcoplasmic reticulum to release calcium. So it releases calcium ions from the sarcoplasmic reticulum, that's these red little dots. And the the calcium will bind to a protein complex called troponin. So these little rounded squares are the troponin, and the troponin is connected to each other kind of with a rope-like filament called tropomyosin. So we have a troponin-tropomyosin complex that is protein. So when the calcium binds to the troponin, it triggers that protein to shift in shape. So proteins tend to change shape when they are bound to by another molecule, and when that molecule unbinds, they will also change shape. So the calcium binds to the troponin here. That causes a shift in the tropomyosin. What the tropomyosin is doing right now is blocking binding sites on the actin. So if those binding sites are blocked, the myosin heads cannot attach to the actin. If they cannot attach, then contraction can't happen. So when the calcium binds to the troponin, it causes that protein to change shape, And when it changes shape, it exposes the binding sites. So over here, they show what we call a cross bridge. If the binding site is exposed and the myosin heads have a molecule of ATP, so those two things must happen. The binding sites on the actin have to be exposed, and the myosin head has to have a molecule of ATP to use. Remember, ATP is our energy. It's going to allow every function in the body to occur. That's why cellular respiration is so important. So if those two things are true, the myosin head will break down the ATP, it will use it, and break it down into ADP and phosphorus, which causes the myosin head to change shape, and it will bind to the actin. Then the ATP will leave the myosin head, it's been used, and another molecule of ATP will attach. Okay, so this is a constant cycle. But when this cross bridge forms and the myosin head changes shape, it's going to pull towards the center of the sarcomere. If you can track my cursor right now, it's going to change shape and pull the actin towards the center of the sarcomere. If that happens, you are going to see the Z lines of the sarcomere come closer together, and that sarcomere is going to contract. This will put tension. If this happens throughout every sarcomere of the myofibril in the fascicle of the muscle belly, then you're going to create enough force through this small contraction to pull the bones that the muscle is attached to create enough force to move them. All right, so I'm going to go through it quickly one more time. Motor neuron receives the impulse. Acetylcholine binds to the sodium channels. Sodium channels release sodium into the sarcolemma. That sodium triggers calcium to be released from the sarcoplasmic reticulum. The calcium binds to the troponin. That causes a shift in the shape of the proteins. So the troponin and the tropomyosin move, exposing the binding sites on the actin. If the myosin head has a molecule of ATP, it can then attach to the actin, hydrolyze that ATP, change shape, and pull the actin towards the center of the sarcomere, creating that muscle contraction.